Welcome to your lecture on the structure and functions of cells in the nervous system. In the previous lecture, we addressed learning objectives 2.1 to 2.5. In this lecture, we are going to discuss the resting potential, hyperpolarization, depolarization, and the action potential in neurons. We're going to summarize the contributions of diffusion, electrostatic forces, the sodium-potassium pump in the membrane potential phenomenon. And we're going to summarize ion movements as part of the action potential, describe the propagation of an action potential, and also talk about structures and functions of presynaptic versus postsynaptic cells involved in a chain of communication. In the next lecture, we will address learning objectives 2.11 through 2.18 listed here. So now on to the discussion of neural communication, the electrical potentials of axons, the membrane potential, the action potential, and its conduction. In this illustration from your textbook, you see the brain and spinal cord of a woman. You see on the left-hand side the brain and spinal cord, the central nervous system. There is a blue arrow going to a cross-section of the spinal cord where the interneuron and the motor neuron are present. In the third panel, we see her hand, and the fingertip of the hand is touching a hot iron. And there you can see it's labeled dendrites of sensory neuron detect painful stimulus. That's a fancy way of saying she's about to hurt herself on this iron. The axon from the fingertip, now working in the opposite direction, is a sensory neuron that receives pain information. It runs to the interneuron in the spinal cord carrying that pain signal. The interneuron then excites a motor neuron, causing muscular contraction, which is basically what propels the withdrawal response, retracting the hand of this person from the source of their pain. Now, the second illustration, 2.14 from your textbook, illustrates the important role of inhibition. Now, imagine you are working at a 3-star, I think that's the most stars you can get, Michelin restaurant. You are a server and you are about to deliver some exquisitely prepared soup to a customer. You pick up the hot bowl of soup, which is sitting on a tray in the middle of a packed dining room, a very elegant dining room, and it is hot. It is extremely hot. So the sensory neuron, that same sensory neuron in the hand that felt the hot iron in your hand, carries the pain signal through its axons from the hand to the interneuron in the spinal cord. The interneuron in the spinal cord excites the motor neuron, causing a withdrawal reflex of the hand, but at the same time, a neuron has an axon that travels down the spinal cord, and this interneuron inhibits the motor neuron from the withdrawal reflex, preventing the hand from dropping the bowl of soup. So we're able to inhibit our reflexive withdrawal from a hot object under circumstances where the ensuing catastrophe would be greater than the tissue injury that we are about to incur. The action potential is basically the terminology used to describe electrical firing by neurons. At rest, the axon has a negative charge, and that is illustrated here in terms of the resting state at minus 70 millivolts. Hyperpolarization is what happens when the membrane potential increases. So when a neuron goes from more negative to less negative, as you can see by item number 4. Depolarization is what happens when there's a reduction in the membrane potential. When you go closer to 0, and even above and beyond 0, becoming more positive. So the message conducted along the axon happens, in a rapid burst of depolarization that's followed by hyperpolarization. There's a threshold of excitation which has been described in your textbook, and that basically means that a set point must be reached in order to produce the action potential in every individual neuron. Note that your textbook publisher, Pearson, has a number of very useful simulations or tutorials on aspects of the action potential, the resting potential, and forces of diffusion, which we'll turn to now. Diffusion is movement of molecules from areas of higher concentration to areas of lower concentration, leading ultimately to some equilibrium or balance of those molecules. In order to understand the action potential, we need to understand the phenomenon of diffusion on the one hand and the phenomenon of the membrane potential on the other hand. This illustration describes the electrostatic pressure that characterizes neuronal membranes, where we have ions, negatively or positively charged ions on either side of the membrane that are trying to establish equilibrium. So the force of electrostatic pressure on the membrane is caused by cations, positive ions, and anions, negative ions, that attract if there is one negative ion on one side and a positive ion on the other, and repel in the instance that you have on opposing sides of a membrane two positive or two negative ions. So this illustration shows diffusion superimposed on the cell membrane. You can see that in the extracellular fluid, particles, these red dots that are of high concentration, move through the phospholipid bilayer that makes up the membrane, entering the inside of the cell or the cytoplasm, and they do so until they reach an equilibrium. And so it is that the membrane potential and likelihood of a neuron firing depends on what's happening with respect to the, on the one hand, diffusion of anions and cations, and on the other hand, that their balance or the extent to which they exert electrostatic pressure on the membrane. So in this illustration, you see the membrane separating the inside and outside of the cell. The outside of the membrane, the extracellular fluid, is positively charged, and the inside is negatively charged based on the assembly of ions therein. The ions on both sides of the membrane are abbreviated with A, negative, K, positive, Cl or chloride, negative, and Na, positive. So the A is an organic anion, potassium is the K, chloride is the Cl, and sodium, which is salt, is the Na. So the organic anions are on the inside of the cell and they can't leave. The potassium ions are of a low concentration on the outside, and they leave the cell through the force of diffusion and enter the cell through the force of electrostatic pressure. The chloride ions are of high concentration outside the cell and enter the cell through diffusion and exit through electrostatic pressure. The sodium ions are of high concentration outside the cell in the extracellular environment, and they get into the cell through both the diffusion and electrostatic pressure. The membrane potential is controlled by the sodium-potassium pump, which has the job of pumping 2 potassium ions inside the cell and 3 sodium ions outside the cell to keep it balanced. The sodium-potassium pump is made of protein molecules that are embedded in the membrane. It continuously pushes sodium ions out of the axon and thereby consumes 40%, almost half of a neuron's metabolic resources. Be sure to check out the simulations, again, provided by your textbook publisher on the sodium-potassium transporter, and also an overall view of the membrane potential. Here is an illustration of a cell membrane for a neuron. You can see that in purple is depicted the phospholipid bilayer. So the heads are pointing outward or inward, and they are bits of fat, and the water-soluble tails of this bilayer are pointing towards the center. What this means is that unless you're fat soluble like a steroid, which we'll talk about later on in this lecture, you are not going to get into the cell unless you get in through one of these ion channels. So the membrane separates the inside and the outside of the cell. Many ion channels are open, others are closed. Each is like a tube-like structure with a pore in the middle, and that pore is the way that the ion is transported from outside to inside and vice versa. And it's surrounded by some protein subunits. The membrane, again, is made up of lipid molecules in 2 layers. And when an ion channel is closed, the ion movements to and from the membrane are restricted. When the ion channel is open, the ions can be transported in and out of the cell through the pore of the ion channel. So how does this fit in with the action potential? The action potential is caused by the brief increase in permeability of the membrane to salt or sodium, which is Na+, followed by a brief increase in the membrane's permeability to potassium, or K+. There are neural membranes that contain thousands of ion channels. The action potential stems from the opening and closing of ion channels and also the distribution of ions. And these voltage-gated ion channels discussed in the previous slide are an example. Figure 2.19 of your textbook finally shows us what is happening simultaneously with respect to the action potential and the opening or closing of ion channels in the neuron membrane. So there are 5 steps, and we're focusing on the first part of this image right now, which I have highlighted with the green oval. There are 5 steps in terms of what's happening with regard to ion transport in the membrane. In the first step, which is labeled open, and mind you, I want to take a moment to note the ion channels look green when there's sodium passing through, and they look blue-ish when there's potassium passing through. So in the first step called open, the ion channel is open and the sodium enters the ion. The outside of the cell, as you can see by the arrangement of pluses and minuses on either side of the cell, the outside is positive and the inside is negative to both left and right of the ion channel, because we have, remember, negative and positive ions on either side of the membrane exerting that electrostatic pressure. In the second step, which is labeled open here, the ion channel is open and the potassium ion leaves the cell. The outside of the cell is positive and the inside is negative to both left and right of the ion channel again. And the ion channel is that green or red entity. Green or blue entity. During the third step, which is labeled refractory, the outside of the cell is negatively charged and the inside is positively charged only to the left of the ion channel. During the fourth step, which is labeled open, the ion channel is open and the potassium leaves the cell. The outside now is negative to the left of the ion channel and positive to its right. The inside of the cell is positive to the left and negative to the right of the ion channel as well. In the fifth and final step, which is called the reset step, two ion channels can be seen side by side. The sodium ion enters the cell in the ion channel on the left. The ion channel on the right appears closed. The outside of the cell is positive and the inside is negative to both left and right of the ion channel. Now that we have looked more closely at ion movements across the membrane, we're going to look at what's happening with regard to the electrical, the nerve impulse, the action potential, focusing on the bottom part of this illustration where I have a big green arrow pointing. We can now see that the same 5 steps discussed above relate to what's happening in terms of the electrical charge. So the line starts in this graph. Let me just take a step back and say that the y-axis shows the membrane potential in millivolts from -70 to +40, right? The x-axis, by contrast, is that horizontal line, and it goes from -70 on the y-axis all the way across. So essentially showing you what is happening at and then below baseline when we get to step 6. But the line starts flat at the threshold and makes a large peak and then falls below the threshold. And that's where we get the step 6 below the -70 on the x-axis. In step 1, we have the start of the peak at approximately -60 millivolts on the y-axis. It is during this step that the sodium ion channels are opening and sodium begins to enter the cell, as you could see from the image above where sodium is entering through the green ion channel. In step 2, we're at the middle of the rising peak and the line is climbing. We're at approximately -20 millivolts, which is more positive, right, than -70 on the y-axis. During this step, the potassium ion channels open and potassium begins to leave the cell. In step 3, we're at the top or the peak of the electrical surge at approximately 40 millivolts plus 40 on the y-axis. So that's above and beyond the negative 70 we started at. And during this step, the sodium ion channels become refractory, as you see from the illustration of what's happening at the membrane, which we discussed moments ago. And so no more sodium ions enter the cell during this refractory period. At step 4, we're near 0 on the y-axis, and the line is steeply sloping downward and downward. And during this step, potassium ions continue to leave the cell, and they cause the membrane potential to return to rest. Step 5 is near negative 60 on the y-axis. During this step, potassium ion channels close and the sodium ion channels get reset. Finally, at step 6, which I alluded to when I mentioned the horizontal x-axis, it's below the threshold, and during this step, the extra potassium ions outside diffuse away from the cell. So with the action potential, we have the inside of a neuron becoming more positive relative to the outside of the neuron, going from minus 70 millivolts to 40 millivolts on that y-axis. And at that time, the sodium ion channels become refractory and no more sodium ions enter the cell. So you get a nerve impulse, an action potential that then is conducted from the soma all the way down the axon, and that's what we call axonal conduction. And the action potential travels along the axon. It stays consistent or constant in size and force. Even if the axon branches, it doesn't get any smaller. And again, it travels in one direction in keeping with the all or none law and the rate law. So what is the all or none law? As you'll remember from an earlier slide, the depolarizing stimulus is what causes the action potential. This illustration shows the axon, which is the sort of orange brownish cylinder or tube, and these microelectrodes that are monitoring electricity as it goes from one end to the axon to the other end. So from the soma end to the end that's actually projecting onto the terminal buttons onto the postsynaptic neuron. So the action potential travels from left to right, and the depolarizing stimulus acts somewhere, in the vicinity of the left, maybe on the dendrites or on the soma, and it shows, based on these 3 electrodes that are monitoring the action potential as it travels, you can see that the amplitude, the amount of electricity that's flowing is constant throughout the passage along the course of this axon. So what is the rate law? The rate of axonal firing of the action potential is really what codes information about the source of the depolarization. So in this illustration, you can see how much firing happens in response to a weak stimulus, such as a candle, versus a strong stimulus, such as a flashlight. In the first image, the stimulus is on for 1 second, turned off, and during that 1 second of illumination by candle, a total of 9 action potentials is produced. by the neuron. By contrast, with the second image where we have a flashlight that's on for 1 second and then turned off, this is a stronger stimulus. And during that 1 second, lots of action potentials are produced. When we talk about sensation and perception, this will actually make a lot of sense because you'll see that loudness and brightness, the intensity of sound, the intensity of light, is something that is detectable even among people who are hard of hearing or visually impaired. This illustration shows the importance of myelination. You can see that where we have the translucent blue and there's myelin, the action potential is much stronger than where we don't have myelination, basically in those nodes of Ranvier, right, between sheaths of myelin. So this is an important point because it means that when a depolarizing stimulus that will trigger an action potential is delivered to the axon, in this instance by a microelectrode, the arrow flows from left to right show that as the action potential is traveling through the myelin sheath, its conduction decreases slightly and it's regenerated again at the nodes of Ranvier. For more on the action potential, check out these other simulations by your textbook publisher on the Pearson website.